All right, guys, in this video, we're going to make sense of photosynthesis. It's a topic that can be complicated sometimes, but it really doesn't have to be as bad as most people make it out to be. So just to set up the idea, remember, photosynthesis is happening in any organism that we call a photoautotroph. That includes plants and algae and some bacteria, and all it really means is they are making their organic compounds, meaning carbon-containing things, using energy from sunlight. That's all it means. There are a different category of organisms called chemoautotrophs that instead of using energy from sunlight, they get their energy from other chemical compounds in their environment. These are fairly rare. You're seeing these in hydrothermal vents at the bottom of the ocean. The vast majority of autotrophs are photoautotrophs, and so it's sunlight that we're interested in. So the way this is all happening, remember, a chloroplast is just an organelle within any plant or algae or bacterial species that is making these reactions happen, right? And so the big picture is, inside of a chloroplast, we have these repeating segments, stacks of cell membrane, almost, that are called grana. And in the grana, each individual section is one thylakoid. So that's a thylakoid, that's another one, that's another one. Stack them all together, and you get grana. Now, they're not really just pure sheets of membrane. They're almost like discs that have a hollow space in the middle. Right? And so the membrane, the thylakoid membrane, wraps around the exterior, and then the space in the middle that they're highlighting in red on here, we would call that the thylakoid lumen. Remember, lumen just means any open space. Mitochondria have a lumen. In the same way, these chloroplasts have a lumen. Right? And so within these membranes, these stacks of membrane, that is where the photosynthesis reactions are happening. And the reason that we put them in these stacks of many thylakoids piled on top of each other is it gets us a lot of surface area, right? More surface area, more membrane, more of the enzymes and transporters that actually make photosynthesis happen, right? Now, the overall reaction that's taking place is plants are taking in carbon dioxide and water. These are reactants to our process. And remember, CO2, right, this is low energy carbon. This is what was given off by cellular respiration, right? So when you had cellular respiration in a human or an animal or any organism that has to eat its food, we let out the CO2 and water as waste, right? Plants are taking that low energy carbon and CO2. They're using the energy from sunlight to drive this endergonic reaction. Right, this would be endergonic, meaning it's not spontaneous. It doesn't happen all by itself. You have to put energy into it. That's the role of sunlight. And what do? You take the carbon and oxygen and hydrogen from CO2 and water, and you build sugars. Right? This is glucose or fructose, any of those monosaccharide sugars. And the plant can use those for whatever it wants, right? Building starch or cellulose, right? The polymers of carbohydrates that you talked about in a previous section, right? At the end of all this, we have a few oxygens left over, and so oxygen as O2 is the other waste product. Remember, that sugar plus oxygen, when organisms that have to eat their food, heterotrophs, like a human, take in the sugar, breathe in the O2, we're just doing the reaction in reverse, right, through cellular respiration, converting it back into CO2 and water, extracting energy along the way, right, that we use to make ATP, and so the cycle just continues repeatedly. Right? Plants use sunlight to make sugars and oxygen. Heterotrophs, like animals and humans, take in those sugars and oxygen molecules, extract the energy back out, and send the CO2 and water right back around to another plant to repeat the process all over again. Right. So, next thing we have to do is break this down into two parts. Photosynthesis actually consists of two different sets of reactions. The light-dependent reactions, which, exactly like they sound, they directly require the input of sunlight. And this is where we're taking water molecules and we're stripping off their electrons, right? Because that's where the energy is. The light excites those electrons, and that's how we're going to extract the energy to make a couple different things. We're going to make ATP and NADPH, right? These are our energy carriers, right, that are going to take that energy that we got from sunlight, and we're going to send it over to something called the Calvin cycle, which involves actually taking CO2 out of the atmosphere and using the carbon for our CO2 to make this Ga3P molecule, which is the building block that we'll then use to make glucose. Right, so the end goal is convert CO2 into glucose, but in order to do that, we need an energy source. Right, so light dependent harvests the energy from sunlight, converts it temporarily into ATP and NADPH, and then we use those high energy molecules to force CO2 to turn into carbs like glucose. Right, otherwise, this process wouldn't happen unless we put a lot of energy in to make it happen. Right, so where does chlorophyll come in? Chlorophyll is really just the pigment molecule in chloroplasts that is absorbing the light energy. So when you see this electromagnetic spectrum, these are your chlorophyll molecules. There are different types, and all they're doing is absorbing light that hits the plant. So what you notice is these lines, these peaks, are just showing you absorption. Think of absorption as energy capture. This is basically asking what percentage of energy can these molecules capture from sunlight. Okay? And so chlorophyll A, as an example, has a peak here, right, in the low to mid 400s. That's blue light. And it has another peak here in the mid 600s. That's red light. And so if you shine either red or blue light on chlorophyll A, it is very good at extracting that energy. And chlorophylls B and beta carotene are slightly shifted, but they're similar. And so these chlorophyll molecules, if you shine blue or sort of purplish, dark green light on them, they're very good at extracting that energy. If you shine red light or orange light, same deal. But this lighter green, yellow region, not so good. And this makes sense because the reason that most plants appear to be this color is because that's the wavelength of light that's being reflected back off the plant because it didn't get absorbed and used for energy. And so counterintuitively, green light isn't actually that good at stimulating photosynthesis. It gets reflected back off. That's why it hits your eyes. Blue and red light, much better. All right, so this is where things get a little complicated. The first part of this, these light-dependent reactions, so give yourself some context. This membrane, that is the thylakoid membrane, and this is the thylakoid space. All right, so when we saw all those stacks of thylakoids, we're inside one of those stacks at this point. And the way that this is all starting off is when light hits this first molecule, right, and these big purple blobs are proteins that are embedded in the thylakoid membrane. When light hits that first protein, what it's able to do is take water, and we're literally splitting the water molecule apart, taking its electrons, and the electrons are boosted to a high energy state by the energy from light. Right? Now, the leftovers from that water are oxygen and protons, H+. Right? The protons will follow in a little bit. They're going to become useful. The oxygen is just going to get released into the atmosphere. That's the oxygen that humans and animals and heterotrophs breathe and use for cellular respiration. Right? So the next part of this, we're just following these electrons. Right? They were originally low energy on water. We boosted them to a high energy state using sunlight. And now this photosystem 2, which I realize is completely backwards. Photosystem 2 comes first and photosystem 1 comes second. It's just an unfortunate way that it was named. Those electrons have a high energy state, and they're going to get passed down an electron transport chain, right, from one molecule to the next. Right? These cytochrome complexes, those are the molecules in the thylakoid membrane that are passing the electrons down. And as that happens, some of those molecules, like this PQ, can take protons, right, move them into the space, and in that process, we are concentrating protons in that thylakoid space, right, in the lumen. So we expect to have a very high concentration of protons in the thylakoid space. That's a good thing because just like in mitochondria in humans and animals, we have this ATP synthase molecule. So just like you saw in mitochondria, those protons want to flow from high to low concentration. Right? High concentration here because we're pumping them in, low concentration over here. Right? That drives the connection between ADP and a phosphate and poof, we wind up with ATP again. Right? So thinking back to our overall reaction, remember, we sent two things into the Calvin cycle. One of them is ATP. This is how it's being made. Right? So summary, photosystem 2 takes the electrons from water that were excited by sunlight, raises those electrons to a high energy state, uses the energy from those electrons to pump protons, right, H+, into the thylakoid space, and those protons then flow down their concentration gradient, high to low, across the thylakoid membrane, and that is what drives ATP synthesis. Right. But we still have our electron, right? And so at the end of photosystem 2, right here, you get sunlight again, so another photon of sunlight hits those electrons, boosts them up to a higher energy level again, and now they get passed down photosystem 1 molecules. Right? So these guys are photosystem 1 proteins still in the thylakoid membrane. The difference is this electron is now handed off directly to a molecule called NADP+, and the product of that is we wind up with NADPH. Think of NADPH as an electron carrier, which just means it is transporting that high energy electron. Remember, it's got energy because we boosted it with sunlight. And NADPH just carries that electron into the Calvin cycle, right? So the Calvin cycle is going to take our NADPH from photosystem 1. It's going to take our ATP from photosystem 2, and it is going to use them as reactants, right, to fuel the conversion of CO2 into sugar. That's the whole point of the Calvin cycle, right? So as this is all running using light as an energy source, the Calvin cycle is happening in the stroma of the chloroplast, which is just the liquid that surrounds the thylakoid. So we're not really in the thylakoid anymore, right? We're in the surrounding stroma, but still in the chloroplast. And so what's happening here is we're taking this CO2, right? Remember, CO2 is a low energy molecule. There's not really a whole lot there that's useful. And so what we're doing is taking in CO2, combining it with this RUBP molecule. So kind of like the Krebs cycle or citric acid cycle in mitochondria, it goes around in a circle because our starting material, RUBP, if you pay careful attention, this is a 5-carbon molecule, right? And we're putting in 3 CO2s. So 3 carbons are coming in. And when we combine those, we have a total between these products of 8 carbons. 5 from RUBP, 3 from CO2. As we move through, we're going to take these relatively low energy molecules and we're going to use ATP, remember that came from photosystem 2, and we're going to use NADPH from photosystem 1 to rearrange those carbons and produce a molecule called G3P. G3P is a 3-carbon molecule. So what did we do? We took our 5-carbon RUBP, 3 carbons from 3 CO2s, we rearranged them, and now one of those G3Ps can take those 3 carbons and we can use the G3P to make glucose. Right now, glucose is a 6-carbon molecule, so you'll have to have 2 G3Ps to produce 1 glucose. Right now, plant has glucose to use to make starch or cellulose or to use as an energy source later. Right now, we made 6 of those G3Ps total per turn. The other 5 of them, right, this 5 is telling you that you have 5 G3Ps. 5 molecules times 3 carbons each equals 15 carbons total. We then convert those G3P molecules back into RUBP. And RUBP, we have 3 molecules that are 5 carbons each, right? 3 times 5 is still 15. That's why it's a cycle, right? You put 3 carbons in through 3 CO2s, you take 3 carbons out through G3P, and then the remaining carbons just get recycled right back into RUBP again to start the cycle over, right? The only other thing to maybe add here is this Rubisco, that is just the enzyme that makes this first step happen. So Rubisco is the enzyme that combines CO2 and RUBP to form these first intermediates, the 3-phosphoglycerates, right? You might see just trivia facts on an exam sometime. Rubisco is by far the most common enzyme in the entire world. Pretty much every plant has it. It's absolutely necessary for photosynthesis. And so this is really, this rubisco enzyme complex is where pretty much all the sugar in the world is being produced. All right, last thing to take care of, big picture perspective, right? In our chloroplast, we just did all this. We're putting in energy from light, carbon from CO2. We're using that light energy to make ATP, and they don't show it here, but also NADPH. We use the energy to convert CO2 into sugars, right, like glucose or fructose. And those sugars can either be used to form polymers like starch or cellulose, or, remember, plants also have mitochondria. The plants can use that sugar as a way to store their energy and save it for later. And plants can import those sugars into mitochondria and use them, right, to make ATP to carry out cellular functions too. Or those sugars could be eaten by, an animal or some other heterotroph. Remember, heterotroph just means an organism that has to eat their food. So, like a human, you could eat those sugars. You could produce carbon dioxide and water as a waste product, and that CO2 would just get recycled right back to plants throughout the world that take that CO2 out of the atmosphere and use it to start this process all over again. All right, I hope this was helpful. Follow along on the playlist. I'm going in order of chapters in the OpenStax book, so there'll be many more videos to come. I also subscribe to the channel. That makes sure that you'll get the updates whenever I release them, and they should be coming out pretty quickly.